So, a brief overview of protein synthesis. And chapter 17. Protein synthesis is basically DNA code, construct a messenger RNA from that. That process is called transcription. Take the messenger RNA code, this is found in the nucleus, travel to the ribosome, translation into a polypeptide, packaged at the Golgi apparatus into a protein. A polypeptide is, remember, just a chain of amino acids. Okay, so I want to go through just a little bit of detail of transcription, some things that happen, and then translation. Transcription. Here's a piece of DNA code. DNA is found in the nucleus, and we're not going to get into this right now, but DNA is on a chromosome. A chromosome is DNA wrapped around things called histones. Whoops. Coiled up. It's another histone. Wrap the DNA around it. Coiled up to fit in the nucleus. So wherever we want to look at a gene, let's say we need to express insulin. Your cell needs to make insulin. So the DNA is unwound that side. One side of the DNA is meaningful, the other side of the DNA is not. Your cell also makes a structure, this is DNA. Your cell also has a nucleic acid called RNA. RNA is much like DNA except for these three ways. Has one strand. It has uracil instead of thymine. So U, not T. Comes in three varieties. M for messenger RNA, T for transfer RNA, and R for ribosomal RNA. So in the nucleus of the cell, transcription, RNA is constructed from one side of DNA. And it does it in the same way DNA pairs up. Base pairing, except since RNA has uracil instead of thymine, a U pairs with the A G pairs with the C, A, U, C, C, G, U, A. Now we have constructed a piece of mRNA. The mRNA can travel. So if we were in a cell, this is taking place in the nucleus. This messenger RNA is going to leave the nucleus. But before it does, in order to leave the nucleus, it has to have a cap called a 5' cap on its 5' end. The cell attaches a little sort of protective hat, if you will. The cell also adds what's called a poly A tail. a multiple adenine tail. Not sure why it does that, but it does that as it leaves the nucleus. So the RNA leaves the nucleus. And just a minute here. Let me get my code down, UGA, UCC, GUA. This travels out of the nucleus to the ribosome. Now, this code is essentially the same as the DNA code. Slightly different, but it's the reverse, if you will. So it travels out to the ribosome, and at the ribosome, translation occurs. Now, there's some great gory detail about this that you're going to have to know for your poster, and you're going to have to do a little bit of research about this on your own. But in general, every 3 bases on a messenger RNA codes for 1 amino acid. So this piece of messenger RNA codes for 1, 2, 3 amino acids. Each one of these 3 base things is called a codon. It's a codon, code for 1 amino acid. So at the ribosome's job is to match up. So in translation, we match the correct amino acid with its codon. How? Well, there's a molecule called tRNA. tRNA has a structure. I'm just going to draw it like an L. It doesn't matter. At the end of the L, there are 3 bases called the anticodon. The tRNA also has, on the other end, a specific amino acid. This tRNA will only carry this specific amino acid. So the tRNA that says ACU at this end will match up here. ACU matches with UGA. And it carries a specific amino acid. In every biology textbook invented, there's a chart that says what amino acid goes with what code. Okay? So tRNA's job then is to carry the amino acid to the ribosome. Drop it off. Sort of like a taxi service. And so this chain, and I'm not sure, I might have screwed up here and made a weird code, but it doesn't matter. This chain will form, if I go to the next one, UGA, UCC, GUA. There should be a tripeptide or a 3 amino acid chain that is coded for by that. And if you look at the chart in your book, you can see what that is. Now, I may have screwed up because this may be actually a stop code. The protein is done when it reaches a stop code. And so you'll have this long chain of messenger RNA, and the very last base, the very last codon, is a stop code. That means to stop producing the protein. Now, one other thing to talk about. If this is a piece of DNA with a gene, it's a gene. Okay, so there could be 10,000 A's, T's, G's, and C's. For some reason, in human DNA, parts of this gene are meaningless. So when a messenger RNA is made, that codes for that gene. The meaningless parts have to be removed. Okay, this is called processing. The meaningless parts are cut out. So let's say right here is a meaningless section. So this part will have to be cut out. And maybe down here is a meaningless section. So this part will have to be cut out. That is the job of something called a SNRP. I can't remember the exact name of this, but SNRPs remove the meaningless pieces. Those meaningless pieces are called introns. The meaningful pieces, so I'm going to blacken in here, intron, intron. And when you cut those out, you have left a shorter piece of messenger RNA. Remember, we have to add a cap and a whole bunch of A's tail. And what you have left are what are called the exons, or the expressed parts. So that is protein synthesis in brief.